Hi everyone and welcome to the continuation of Mendelian genetics. Again, I apologize for the length of this video and the previous part. It's quite a bit longer than some of the other lectures because there are a lot of calculations and I like to go step by step just to make it a little clearer. So continuing on with those calculations and probability, next I'm going to show you a way to use probability calculations to predict particular combinations of events in genetics. So one of the ways that we can use it is I can tell you that a certain number of children are born to the parents and then ask you the probability or to predict the combination of, let's say, a certain number of sons versus some certain number of daughters. And sometimes the question will not have both represented. I could simply say there are 6 children. What's the probability of having 2 sons? And then you would have to know, oh, okay, if 2 are sons, then the remaining 4 would be daughters. And we're going to see that in a moment in the example calculations. We can also use these probability calculations to predict events with organism crosses, such as looking for mutant versus wild type Drosophila from a particular cross. And the textbooks or any kind of official websites for this kind of calculation will show the formula in this scary looking way that makes no sense. And I'm going to show you on the next slide how I like to represent it with actual words and get into the habit of knowing exactly what goes where. Okay, so rather than memorizing the random letters of the probability formula, I like to have students get used to writing it out or saying it in terms of what they actually mean. So, for instance, P being probability is equal to the total number of events factorial. So, for instance, if in the previous slide we said 6 children would be born, that's the total number of events would be 6. Factorial simply means you're doing that number times each of the lower numbers. So in that case, 6 factorial is 6 times 5 times 4 times 3 times 2 times 1. And you're going to see this more in the next slide. We then divide it by the number of times of option 1. So for instance, option 1 in the example that we were just doing is sons factorial times the number of times of option 2 factorial, which would be daughter, the number of daughters you want. Then you multiply that fraction by the probability of option 1. So in the case of sons versus daughters, you have 1/2 probability sons, 1/2 probability daughters. So you do the probability of option 1 to the power of the number of times you want that. So if, for instance, you were saying 2 sons, you would do 1/2 probability of getting a son to the second power, okay, because you want 2 of those sons. Then it's the probability of option number 2 to the power of the number of times. So for instance, if the number of daughters you were looking at was 4 daughters, you would say 1/2, which is the probability of getting a daughter, to the 4th power. Okay, and so we're going to go over examples now to make this a little clearer. So let's calculate an example. This first example we're going to do is the probability of finding 5 males and 2 females if 7 flies are born from a cross. Okay, so I'm going to write that here. Probability of 5 males and 2 females if 7 flies are born. Okay, so we're going to use probability to figure out the chances of having a very specific outcome of events, being 5 males and 2 females. So as we saw with our formula before, we do P equals, and what's on top? The total number of events. Well, what's the total number of events here? How many flies are born total? The answer is 7 flies born total.
So you want 7 factorial on top as the total number of events over the number of times option 1. Well, what's the option 1? Males, right? How many males do you want to see? 5 factorial. So that represents males. And then we have option number 2. How many times do you want option number 2? Well, you want 2 females. Okay? Then we multiply by the probability of getting option 1. Well, if you are going to have a fly have an offspring, what's the chances that that offspring will be male, having the XY chromosome? Well, the only 2 options for that fly in this case in terms of chromosomes, again, we're not going into gender identification. This is strictly speaking based on chromosomes for flies. If there's going to be a fly born, it's a 50% chance it'll be male, 50% chance it'll be female, because those are the only 2 options. It's either going to be XY or XX, okay? So it's 1/2 probability of male, 1/2 probability of female. So we would write half, and how many males do we want? 5. And then we write half, which is the probability of it being female. How many females do we want? 2. Okay, and you can kind of remind yourself by writing underneath which option you're talking about, because you have option 1 and option 2. Now, nowadays, you know, it's easy to just plug this into a calculator. Actually, it's not that easy. Students make mistakes with that a lot. Even I do, because one wrong parentheses or whatnot, and it ruins everything. But if you want to figure out what this actually means and you want to try writing out what all of this means, well, let's break this down a little further. So P equals, and we say 7 factorial. What factorial means is you multiply that number by each of the lower subsequent numbers. So 7 factorial actually means 7 times 6 times 5, 4, 3, 2, 1. Okay, and you don't include 0 because if you multiply by 0, the whole thing turns to 0. Okay, now you put that over what's 5 factorial. 5 factorial is 5 times each of the subsequent lower numbers. Okay, and then what is 2 factorial? 2 times 1. Okay, you then have your 2 probabilities. Now, when you have the same fraction for each of those probabilities, you can simply do 1/2 and you add the 2 exponents. So 5 plus, so we would just write 7 up there, okay, to the 7th power. Now, when you have those factorials on top of each other, you can cross out what's the same on top and bottom. So having 5 times 4 times 3 times 2 times 1 on top, we then cross out the 5 times 4 times 3 times 2 times 1 on the bottom. And so you're left with 7 times 6 on top and 2 times 1 on the bottom. Okay, so you can rewrite this as 42 over 2 times 1/2 to the 7th power. And you can break that down even more because 42 over 2 is actually 21. And you could say times 1 over 28. Okay, and you can keep doing this by hand, writing all of this out until you get to 0.164, okay, which again, nowadays you could just take our first formula that we had on the top of the page, plug it into the calculator, and the calculator should tell you 0.164 if you plugged it in correctly. And then to convert that to a percentage, because probability is the percent chance of something happening, how do you convert a decimal to a percentage? You just multiply by 100. Okay, so that's equal to 16.4% chance of having 5 males and 2 female flies born if 7 flies are born in total. Okay, so now I know it made it scary and you probably tuned out when I was doing all of this by hand, but that was just to show you how you could solve this by hand. I will never ask you to do that. For me, I care more about you being able to plug the right numbers into the formula, being able to know offhand without me showing you a formula. When I ask probability of 5 males and 2 females, if 7 flies are born, you should be able to know what is the probability formula and how would you plug those numbers into it. Okay, I won't even make you solve for the final numbers or percentages. You just need to be able to recognize, okay, if I have the probability of this many males, this many females born to this many, total flies, then I'll plug in this number on top, this one bottom, and recognize it in the formula that we see up here on the top. So I put a star by that formula, okay? So the other example that we can do when we do fly crosses, a lot of the times we're talking about the different phenotypes that you could end up with. So for instance, in crosses are the offspring wild type or sepia or wild type or apterous, things like that. So what we're going to do for this example is the probability of 2 flies being apterous out of 5 born. Sorry, erase that. And I'll specify the cross. So out of 5 born to plus A crossed with plus A parents. Okay, now notice in this question there are 5 offspring born, but I only asked you about 2 of the flies being apterous, which means what do you have to not forget to put into your formula? Well, if they're not apterous, what are they going to be? They're going to be wild type, right? So don't forget to leave, to include the rest of the flies, okay? Your top and bottom numbers when you do the formula should add up to each other in the beginning part. I'll show you what I mean right now. So we're going to do P is equal to, what's the total number of flies we're looking at being born? 5. Don't forget to put the factorial. You put that over option number 1, which is 2 flies being apterous, right? So option number 1 is 2 flies factorial being apterous. So what then is our option number 2? If they're not apterous in this cross, they must be, what's the other option? Wild type, right? So if 2 are apterous, the remaining 3 out of 5 is what you want to be wild type. Now notice that the bottom numbers, 2 plus 3, have to equal the top number of 5. That way you know that you included all of the flies, all of the options that we're talking about. Okay? So now, when you have that cross of plus A crossed with plus A, if you do the Punnett square for that, well, you know the probability of having wild type and the probability of apterous. I don't have to draw the Punnett square out for this particular example because you guys already know that anytime you're crossing 2 heterozygous flies in a monohybrid, you expect a 3-to-1 ratio in terms of 3 wild type or 3 out of 4 wild type and 1 out of 4 recessive. So the probability of apterous, because in this case apterous is our option number 1, the probability of having apterous in a plus A cross is 1 out of 4 of those flies in the Punnett square. How many do you want? You want 2 apterous. Then you move on to your next option, which is wild type. And again, I always write the options underneath them so that I know I'm not leaving anything out. What's the probability of wild type in that Punnett square? 3 out of 4 should be wild type. And how many wild type do you want? 3. Okay? If you plug all of this into your calculator, you should then end up with a 26 probability of getting 2 apterous flies, 3 wild type flies in a plus A cross with 5 total flies born. Okay? And we can do this with anything. We could even do this with a dihybrid cross. The only difference with a dihybrid cross is that on the bottom, instead of having 2 options, how many phenotype options do you have in dihybrid? You have 4, right? So if we do this with dihybrid, okay, which I can ask you about, okay, even though we're not going to do a full practice problem with dihybrid, I can ask you about that because you should be able to apply what you learn to go a step further. So I'll move the 26% up here, okay? If it's a dihybrid cross, then you have 4 options that you need to have in your fractions in your probability. You have wild type, you have apterous, you have sepia, and you have sepia apterous, or sometimes again you might see it apterous sepia, as long as both are written out. So you have the double mutant. Okay? And so if you would do that, you would have P equals total on top, and then you would have wild type number factorial, you would have apterous factorial, you would have sepia factorial, and you would have apterous sepia factorial. And then you would have the 4 fractions. Now, where would you get what these 4 fractions are? What do you know in a dihybrid? What is it? It's 9 out of 16 to 3 out of 16, 9 to 3 to 3 to 1, right? When we did that big Punnett square, okay? Sorry, I'm going to erase that so that it doesn't stay messing up your screen, right? So you would have 9/16 is the probability of wild type, 3/16 the first mutant, 3/16 the next mutant, and 1/16 the double mutant, right? This is all stuff that we've covered before and that you've done in lecture. And then you would just have the number of wild type, number of apterous, number of sepia, and number of double mutants. Okay? So you can be asked to calculate probability for monohybrids, for sons versus daughters of flies, and even for the trickier dihybrid cross.
Okay, so now we're going to move into chi-squared analysis, which is very important in statistics and data analysis. This is going to statistically determine how fit your data is. And what that means is you can figure out how close to expected and expected values your data may be. You will start with a null hypothesis whenever you do chi-squared, and we represent it with that symbol. And we always write the same thing for the null hypothesis, which is There is no significant difference between expected and observed results. And you can always specify what the expected results are. For instance, if it was a chi-squared analysis of a monohybrid cross, you can say the null hypothesis is there is no significant difference between expected results and observed results, with the expected being a 3-to-1 ratio. Now, when you do this analysis, you will obtain a chi-squared value using the formula here, and I'm going to go through in the upcoming slides exactly how to use that. This is summation sign, and this is observed minus expected squared over expected. And again, this will become clearer in the next slides where I go through step by step and the examples of this. You then use that chi-squared value to obtain a p-value, and that p-value will then tell you if you can reject or fail to reject your null hypothesis. And again, we're going to go through that now with our example, helping to make it clearer. So I'm going to show you 5 easy steps to tackle the chi-squared analysis. And we're going to use the problem, set up an apterous monohybrid cross of F1 heterozygous flies. And when you count the offspring, you see 250 wild-type flies and 150 apterous flies. So we're going to go step by step to do a full chi-square analysis.
So when we talk about chi-squared, there's a few terms that you need to know that kind of help ease you into the practice problems. The first one you hear a lot if you've ever taken biostats or any kind of statistics class, and that is the null hypothesis. And whenever you're doing any of these crosses in class, you would normally kind of start off with a null hypothesis. Now, I know a lot of times you're used to hearing hypothesis, but you kind of may not have heard this term before. Now, I want to circle this right here. This is H with the subscript 0. So it's H0, null hypothesis is what this stands for. It's not saying HO, okay, just in case you see it in writing elsewhere, okay. Now, when we have a null hypothesis, the null hypothesis is generally always the same. What it is saying there is no significant difference between expected and observed results, okay, in your experiment. So there's no significant difference between expected and observed results. So whenever I ask for null hypothesis, you would say, oh, the null hypothesis is that there is no significant difference between expected and observed results. And you can even then specify what those expected and observed results are. Now, we're going to go through this when we do the practice problems, but I do want to point out an important point to make is when you see expected results for these crosses, well, how do you get what you're expecting? By doing Punnett squares, right? Okay, so you get expected from the Punnett squares that you know how to do. Now, once you have your null hypothesis and you're kind of starting to think about expected and observed results, you then move on to the chi-squared. And the purpose of that is to use your chi-squared formula to get what's called a p-value. Now, the p-value is important because the p-value will then tell you whether to reject or fail to reject your null hypothesis. Sorry, it reflexively wants to erase when you do that symbol with the touchscreen pen. So now notice the p-value is saying that the p-value that you obtain will tell you whether to reject or fail to reject the null hypothesis. What is it not telling you? The p-value will not tell you to accept the null hypothesis. We're just either rejecting or failing to reject that hypothesis. If we needed to, or if we wanted to accept the null hypothesis, we would then have to do further experimentation and tests. Okay? So you formulate, whenever you have to do chi-squared, you first formulate your null hypothesis, then you use a formula to calculate chi-squared, and you use that chi-squared value to get a p-value, which will then tell you whether you can reject or fail to reject that initial null hypothesis you had. Now, when we say chi-squared, what we mean is this formula here. Chi-squared is equal to symbol. Okay, sorry, the touchscreen keeps wanting to automatically delete things. Epsilon symbol, and then we have O minus, sorry, the pen keeps acting up, O minus E squared, and you put that on top of E. And now when we do that, okay, that epsilon, so I'm going to over here put your little key for terminology, okay, that epsilon symbol means sum of, okay, so you could think of that as sum meaning addition, okay. Then O, what do you think O means? Well, in our null hypothesis, we said expected and observed. So it makes sense that O means observed. So the observed number of flies in a category. Okay, so observed is the O is the observed number of flies in a category, and then that leaves you with E. And what do you think E is? Well, we mentioned observed, so now we have to mention the expected number of flies in a category. And again, how do you get that expected? From a Punnett square. Okay, and that's going to make more sense as we go through the practice problems. Now notice that when we wrote epsilon, we said that that means sum or addition, but we only have one fraction written there, right? Well, the sum will be that you do this fraction for each phenotype, okay, when you have a cross. So for instance, in a monohybrid, you have two phenotypes. You have the wild type and the mutant. So you would do that fraction 2 times and add them together. Now we're going to do full examples of this so you can kind of understand it a little better. Before we move on to the examples, there's just one more point I want to make. When you see that O minus E on the top of that fraction, sometimes you'll hear this called deviation, okay? I won't refer to it like that, but sometimes just in case you're reading a textbook or anything, deviation, because you're basically seeing, well, how much did I deviate in my observed phenotypes or observed offspring from what I was expecting? Okay, so we're going to go through an example to kind of help you visualize exactly how to do this. If any of the terms we discussed on this slide is causing you any trouble, you're uncertain, just send me a message and remind, and I will help you out. Okay? So now we said with chi-squared, okay, ultimately we want to come up with a null hypothesis. We want to calculate a chi-squared formula, get that p-value, and then determine whether or not we can reject or fail to reject that null hypothesis. Now notice when we made the null hypothesis, we made it that there is no significant difference between expected and observed results. So basically the whole point of doing this chi-squared analyses is after you've done a cross with the fruit flies, you want to see, did you get what you were expecting? For instance, in monohybrid, you always hear that 3-to-1 ratio, right? Well, how do you know how successful your monohybrid was at the end of your cross? Well, you kind of want to see how close did you get to that 3-to-1 ratio. So that's why the chi-squared, we make that null hypothesis that there's no significant difference between expected and observed results, because ultimately we want to see, was there a difference between expected and observed results? Did we not get the 3-to-1 ratio for that monohybrid or whatever we were expecting? Okay, so if you're going to do this kind of analyses, the first thing you do, you set up your cross with the flies, and then ultimately you get to count those offspring. So in the example we're going to use to tackle chi-squared in 5 easy steps, I know I sound like an infomercial, the example we're going to use is you set up a monohybrid cross in terms of Apterus, looking at the Apterus mutant. Now, when we say that, what kind of flies are we looking at? What is the, Apterus phenotype? What does that actually mean? Well, we're looking at the trait or the characteristic of wings. And Apterus means that we're looking to see, do the flies have no wings? Do they have wings? Okay, so wild type have wings, Apterus do not. So you cross the flies that have the genotype plus A and plus A. Well, what are those? They're heterozygous genotypes, right? And they both look wild type. When you cross those two flies, you then counted offspring as 250 wild type little flies and 150 Apterus flies in the offspring of that cross. So when you mated those two heterozygous wild type flies, you got the birth or the creation of 250 wild type little babies and 150 Apterus little babies. And as you know, they're not actually little babies. They go through the egg and larva and pupa and adult stage, okay? Now, when you want to do chi-squared, the first step you have to do is do the Punnett square to get the expected results, because we already know what we've observed. But now we need to do a Punnett square to get the expected, those E values. Okay, so we're going to look for expected results. So you're all experts at the Punnett squares by now. You make your big square, you put the gametes of each fly on the side. So we said our cross was plus A fly and plus A. So plus A fly and a plus A fly. And this is the standard, sorry, the pen is acting up. So I'll just erase that for you. The standard monohybrid cross. Okay, so you end up with the usual expected phenotype ratios of 3-to-1. Now keep in mind, when we talk about expected and observed results, we're talking about phenotypes. And the phenotypes, you're already experts at knowing what phenotypes are. They're wild type, they're Apterus, and in other crosses, they're things like sepia, white-eyed, yellow-white mini. All of those are what we mean when we say phenotypes. So when we look at this Punnett square, we are expecting, sorry, the pen is acting up again. We are expecting 3/4 of the flies to be wild type and 1/4 of the offspring flies to be apterous, meaning no wings. Now, when we look at our monohybrid cross that we did in the example, you notice that we saw 250 wild type and 150 apterous flies. That means that the total number of offspring that the cross yielded ultimately was 400 flies. And again, the way you got that, in case when you're watching this you don't realize, it's the 250 plus 150 offspring flies. So if we want to know how many offspring we were expecting to be wild type and how many we were expecting to be apterous, all we do is multiply the expected fraction by the total number of flies that were created in this cross. So expected wild type is 3/4 of the 400 total flies. So you were expecting 300 wild type offspring to be born. For the expected apterous, you're expecting 1/4 of the 400 offspring to be apterous. So you are expecting 100 apterous flies, okay? And you could think of this in English mathematics, using the English language and math. Whenever you say, of, notice I said 1/4 of 400, 3/4 of the total offspring. That tells you multiplication, okay? So in order to get expected, you do a Punnett square and you multiply the fraction of each phenotype by the total, okay? So now we get to everyone's favorite step out of the 5 steps because it is the easiest. So you notice that in step 1, we calculated the expected number of offspring for each of our phenotypes. But when we first introduced the idea of chi-squared, you notice it wasn't just expected in that formula. What else was there? The observed number. So step number 2 is to note the observed values for each of your phenotypes in your problem. And so in our problem, we have 2 phenotypes. We have wild type and apterous. And what do you notice? The observed numbers are right in the question itself, okay? So the observed number of wild type from this cross is 250. The observed number of apterous from this cross is 150. Okay, nice and easy step. Now comes the more annoying step, the step that students usually find more annoying, which is to plug in your values into the chi-squared formula. Now, just as a reminder, what did we say the chi-squared formula was? Well, chi-squared equals the sum symbol O minus E squared over E. Okay? So now we're going to plug that in, knowing the expected and the observed values. And like I said, you go by each phenotype. Okay, so the first phenotype we'll do is wild type. Okay, and we say, how many wild type flies did we observe in the offspring? 250. It was right in the question. And how many wild type offspring did Well, we expected 300 because we expected 3/4 of our 400 little baby flies to be wild type. So the observed 250 minus the 300 expected, and we square that, so there's not going to be any ultimately negative numbers, and you put that over the expected, which is 300 again. Now, we said that chi-squared is epsilon, O minus E squared over E. And we said the epsilon symbol means sum, meaning addition. So this first fraction only represents the wild type flies. But in a monohybrid cross, you have two different phenotypes, right? So now you're going to add the values for Apterus. So for the Apterus flies, how many did we observe? 150. It's right in the question. Then we had already calculated the expected to be 100. We square that and we divide by 100 because both of those 100s represent the expected number of Apterus flies. Okay? And then once you do that, you can either, usually use a calculator, or you can draw it out. So you say chi-squared equals, and you draw out that's the 250 minus 300 is minus 50 squared over 300 plus 50 squared over 100. Okay. Ultimately, that should give you 8.33 plus 25, which is equal to 33.3. So now we have our chi-squared because that 33.3 that you just saw is our chi-squared. So now we can move on to step 4. And step 4, you have to determine what your DF is. And DF stands for degrees of freedom. And I know it sounds scary, but all it is the number of phenotypic categories minus 1. And again, remind yourself, what does phenotypic mean? What are your phenotypes? Well, they're things like wild type, Apterus, sepia, okay, those terms that we've already talked about. So there are usually two different types of crosses that you're working with in genetics lab with the chi-squared. It's usually your monohybrid and your dihybrid. So what would the DF be in a monohybrid cross? Well, think about it. How many phenotypes do you have possible in the offspring of any monohybrid cross? You have 2, right? Either wild type or mutant. Okay, so that's 2 phenotypes. So for that one, it would be 2 minus 1. So the DF in a monohybrid cross will be 1. In a dihybrid cross, now instead of just having wild type and the one mutant, such as Apterus, possible, think back to our dihybrid cross that we did when we were looking at wings and eyes. Well, we had the offspring could be wild type, they could be just Apterus, they could be just sepia, or they could be the double mutant. So they could be sepia Apterus, okay? Or some people call it Apterus sepia. Either way, there's the wild type, there's individual mutant 1, individual mutant 2, and the double mutant. So in the dihybrid cross, you have 4 possible phenotypes. And so to calculate the DF, you would do 4 possible phenotypes minus 1. So the DF in any dihybrid cross will be 3. The last thing to mention is think about our own example here. So in our example, we had a monohybrid cross, right? We had the phenotypes being wild type and Apterus. So the DF in our example would simply be 1. Okay? If you have any trouble understanding anything, just send me a shout out in the Remind app, please. Okay, so now we get to the final step. I know it took so long to get here, right? You put in so much work, and then you are finally at step number 5. And what step number 5 is, I'm going to switch over to pen mode, is you will use your chi-squared NDF to find the p-value. Okay, so use chi-squared NDF to find p-value. And what p-value means is the probability value. Now, we already in our previous steps calculated that the chi-squared in our example is 33.3, and the df in our example is equal to 1, okay? So what we would do is you're always given a table, and on the table, you will try and find your p-value based on the chi-squared and the df. Now, I want to point out when you're using the p-value table, all of these numbers here are df values. So the ones that you're usually going to be using are either the 1 or the 3, because 1 is the df for monohybrid, 3 is the df for a dihybrid. Then all of the numbers up here that I'm circling right now, these decimal numbers that are all lower than 1, those are the p-values. And notice the p-value always has to be lower than 1. And I'll explain that in a minute when we kind of relate the p-value to being sort of like a percentage. So when you see 0.995, that's like 99.5% probability. And now if you had a p-value of 1, that's like saying 100% probability. You can't go higher than 100%, okay? So you won't see a p-value higher than 1. So we see our df, we see our p-value, and then all of these numbers in the center, all of those are chi-squared values. Okay, so when you want to use this table, the first thing you do is you find your df, which we said is 1, so we're looking there, and then you scroll across until you make it to where 33.3 would be. And what do you notice? The chi-squared values are getting bigger, but we can't find 33.3. So what does that tell you? That tells you that your p-value would be off the chart in this direction, 33.3. Shows you that, sorry it cut off, but you know that I wrote 33.3. That tells you that if you're then looking up to see what p-value it matches with, it tells you your p-value is off the chart as well. Now, what do you notice? The chi-square values, as you went left to right, the chi-square values got bigger. What do you notice about the p-values as you're going left all the way to the right? Well, we start with 0.995 over here, but as we go across, they're getting smaller. So if you're looking at a p-value that's off the chart to the right here, that tells you that it would be less than the last number that you're seeing. Okay, so our p-value for this example would be less than that last number that we saw, which is 0.005. Okay, last number of the p-values that I'm circling here. Our number would be to the right of that, which means less than 0.005. Okay? Now, when you use this chart, there's a very important number that I want you to keep in mind in statistics, and that is a p-value of 0.05. So whenever I look at one of these charts, the first thing I do is I put a big red square around the p-value of 0.05 and all of the chi-squares that correspond with that, okay? The reason that number is so important is that when you end up with a p-value less than 0.05 and less than, remember, p-values get smaller as we go to the right. So this would be p-values less than 0.05, okay, any of those, then you would reject your null hypothesis. Okay, so reject null hypothesis. Anything greater, so p-values greater than 0.05, which is all the values to the left of that 0.05, then you fail to reject. Now notice we said before you're not accepting, you are failing to reject the null hypothesis when your p-values are above 0.05. Now keep in mind, in our example, our p-value was less than 0.05. So we rejected the null hypothesis. So what does that mean? That means we reject the idea that there is no significant difference between expected and observed values. So if you are rejecting the idea that there's no difference between what you saw and what you were expecting, if you're rejecting that, what does that mean? That means that there likely was a difference between expected and observed. And if you look at our numbers in the examples, you can see that, you didn't get 300 and 100. You got quite a bit different from that. Okay, so that's kind of how you can kind of think of the final result when you're analyzing your p-values. Okay, so in step 5, make sure you always remind yourself what your chi-squared was, what your df is, and then you look up where your chi-squared would fall on this table. And then wherever your chi-squared is, you go to the top of that column and you ask yourself, okay, that column at the top, the p-value is this value. And then the final step in any chi-squared, you always want to ask yourself, is my p-value less than 0.05 or greater than? Okay, so I want you to imprint that number in your head, p-value 0.05. If your p-value is less than that, you reject the null hypothesis. There's quite a difference likely between your expected and observed. And if your p-value is greater than 0.05, you fail to reject your null hypothesis, and odds are what you got looks very similar to what you were expecting. Okay? If there's any questions, if you're having trouble reading the chart or understanding what we did on here, because I know, it's not as easy to see maybe as if we were in the classroom. So just send me a remind message, take a picture of the slide that's giving you trouble, and I'll try to clarify it step by step. Okay? So now that's it for calculating the chi-squared and analyzing it. You have the 5 easy steps. I just want to give you a little bit more information on kind of how you can think of the p-value and kind of why less than 0.05 is rejecting the null and considered significant. So when you think of the p-value, I want you to think of p-value as a percent, okay? Even though p stands for probability, you can think of it as percentage. And basically what this means is if you repeated the same experiment, the same cross, several times and your p-value is 0.25, then you could think of it as, okay, 25% of the trials would exhibit chance deviations from the initial trial. And when I say chance, that means that any deviation you're seeing is simply by chance, okay? Whereas a p-value of less than 0.05 means that the probability is less than 5% that basically less than 5% chance that the observed deviations are from chance alone. Okay, so if it's saying that there's less than a 5% probability that you're seeing kind of deviations from expected based on just chance, that means something else is at play, okay? That means that there's a reason for the difference, and that tells us that the deviations are significant. Now, I want you to circle, star, highlight the term significant, okay? Because when we do chi-squared, anytime the p-value is less than 0.05, we say that the results are significant. Okay, so you'll see in publications, in scientific papers, that's when they say there's a significant difference between, expected and observed. The reason they're allowed to say significant is because they did the statistics and the p-value is less than 0.05. Okay, you don't want to, in the sciences, say significant if you haven't done the analyses and seen a p-value less than 5%. Okay, so I know that that's kind of sometimes a little tough to kind of think about, what the p-value actually means. I'm not going to test you on the concept of repeating several times and what's by chance, what's not. I'm going to test you on doing the chi-squared and actually analyzing, your results of a cross based on that. But on this slide, what you do need to know is the term significant, and make note that if a p-value is less than 0.5, you then use the term significant difference, okay?
So whenever you're doing a chi-square test or you're looking at a p-value table, just remember that when the p-value is less than 0.05, then the result is considered significant and you reject the null hypothesis. The last topic that I want to introduce briefly is the use of pedigree charts or pedigree genetics to predict and to better analyze genetic inheritance. It basically looks at inheritance in humans to predict how a trait is inherited or carried down throughout generations. Now, please note that the terminology that they use in these pedigree charts is related to chromosomal inheritance, not identity. Okay, so you will see female and male, and sometimes sex unknown, but we don't see non-binary represented here because they're looking at chromosomal inheritance rather than gender identity. So that's a completely different topic. What's valuable about using these pedigree genetics, and yes, I love the gummy bear genetics. Every time I see this slide, I am practically drooling. But yeah, when we use pedigree genetics, it's very valuable to be able to predict and analyze whether a disorder is autosomal dominant, autosomal recessive, or has X-linked inheritance. And again, X-linked can also be dominant versus recessive. So I'm just going to show you some of the kind of examples of what you would look like if you have autosomal traits, and then next we'll look at X-linked traits. The way to tell kind of quickly, just eyeballing a pedigree chart as to whether or not it's recessive or dominant is going to be to look if it skips any generations. So the moment that you see that a trait skips a generation, okay, because notice if the color is filled in, that person has the trait showing. If the shape is empty or not colored in, that represents someone who is not showing the trait. So sometimes they say, healthy versus unhealthy, but it's sometimes just a matter of do you see the trait? Do you not see the trait? Okay. And so when we look at this, the moment that you see that there's a parent who has the trait, but then the offspring do not have it, and then it pops up again in the next generation, that's usually a clue that that is a recessive trait, whereas dominant traits, you see them go from one parent to an offspring to the next generation. So you see all the generations here are showing that trait. Now, you're then going to want to see, is it autosomal or is it X-linked, or sometimes they call that sex-linked, meaning that the allele for that trait is on the X chromosome, and suddenly the gender of the organism will matter in terms of inheritance. Well, the way to tell that is if you see one gender more inflicted, that will sometimes give you a clue. So you'll notice that, sometimes they're kind of evenly spread out, sometimes they are not, but there are certain patterns that will kind of show us X-linked inheritance. So I'm going to skip to the next slide so that you can see what X-linked really shows on a pedigree chart. So now with X-linked inheritance, we're going to look for some patterns. First of all, in X-linked inheritance, because males are getting the Y chromosome from their dad, and they're not going to be able to get an X chromosome from dad, what happens is you will not see any males getting the infliction or the trait from their dad. Okay? Now, one of the ways that you can look to see dominant versus recessive in X-linked, in dominant, you'll notice that a male will give, he only has one X. If he's infected, that means that one X is carrying the infected allele. And where does he give that X? He gives it to his daughters. And if it's dominant, that means that the daughters who automatically are getting the only one X from that infected dad, they will have the trait. So in X-linked dominant, if you see a square, the squares are colored in, always have the females colored in, their daughters have the trait that's X-linked dominant. In X-linked recessive, the other way that you see the inheritance happening is the opposite. So in X-linked dominant, dad has to give to daughters. So you can circle the D of X-linked dominant and remind yourself dad to daughter. Whereas X-linked recessive inheritance, if a trait's recessive and on the X, then females who are XX that have the trait because it's recessive, that means both X's of this female have to have the we'll call it inflicted trait, right? Or the unhealthy trait. XX is unhealthy here. So if she has to give one X to her sons, and the sons are not getting any other X to compensate for that, right? Because they're getting a Y from dad. Okay, so their one and only X that the sons are getting is coming from an infected mom. So you'll notice that moms who are infected in X-linked recessive, they will have each of their sons will also be inflicted with that trait. Okay, so here's another female inflicted. Each of her sons have the trait. Okay, so for X-linked dominant, think dad to daughter. Dads must give the trait to the daughter. In X-linked recessive, moms must give the trait to their sons. Okay, so I know that was a lot of information thrown at you at once. I just want to give you one example of a practice problem. Please send me your answer to this problem right now. Pause the video, send me your answer in the Remind app, and send me a picture of the slide so that I know exactly what you're answering. Now, what I want you to send is the picture of the chart and tell me what kind of inheritance is this shown in this pedigree chart. Is it autosomal or X-linked? And is it dominant or recessive? And remember, the first thing you should be doing is look to see, does it skip a generation? The moment that you can find an inflicted, meaning a colored in offspring that has neither parent colored in, that tells you it has skipped a generation, okay, which would tell you whether or not it's autosomal or X-linked. And then for dominant recessive, You can first look to see, is there one of the genders that's showing it a lot more? Are there a lot of squares colored in, for instance, compared to circles? Or then look for the patterns that we talked about a moment ago. For instance, dad to all the daughters or moms to all the sons. Okay? So send me your answer in the Remind app, please. That is it for today. Again, I'm going to keep apologizing for the length of part one and part two. Thank you for staying tuned in. If you have any questions, please contact me in the Remind app. If you need some more practice with anything, always just send me a picture of the slide that's given you any trouble or of the practice problem you're answering. That way I can help you a little bit better. Okay? Thank you and have a great day.